Wolbachia is being used to control vector mosquitoes, which transmit avian malaria to Hawaii's endangered bird. There's a mouthful. There's me. I present this as Department of Health. Libby Char, the former director, she changed over with the new administration. She allowed me to say this, and I gave it in service to Department of Health in Hawaii. Fine. I'm going to talk specifically about the intervention, on these aspects. There's many aspects, but I'm going to talk about horizontal spread of Wolbachia. Don't call it horizontal gene transfer. That was in genetic modified stuff where the gene spreads. I'm not talking about gene spreading. I'm talking about the germ, Wolbachia, spreading to where it shouldn't belong. Then people said, well, GPAN, all you do is complain. What solution do you have? I'll give you an alternative solution, okay? I think my solution has been overlooked. I don't know if they even considered it. I think it has less impact and it's easier to recall if something goes wrong. I told Tina I wouldn't mess with their math model. I wouldn't criticize what they did. I'm gonna assume it works. But since I finished my part, horizontal gene transfer and alternatives. Oh, by the way, Scott, Tina, you can see my mouse thing as I swirl it around, right, when I try to point.
Yep.
Okay, good. Then I really a little bit concerned about their math model, okay? Specifically, I wanted to see it, but I couldn't find it. And so I read other people who prepared it and what they had assumed and what they didn't. I'll give you a very simple explanation on how math models break down without the math, okay? All right, so here we go. Horizontal germ transfer. Here's a guy using the gas, and here's the collateral damage. It really means when this thing goes beyond what it's supposed to do we have damage? I just told you that the Wolbachias can invade any insect, arthropod, and basic worm. Okay, so here it is. We are trying to wipe out all the mosquitoes by introducing a red male to a gray female. Okay, you remember that basic picture? You introduce red male to gray female and you wipe out all mosquitoes. Mm-hmm. Well, what if something goes wrong? What if there's a red female and red male? Whoa, I thought you weren't supposed to release females. Yeah, well, 3 out of a million. And if you double read it with enhanced technology, 1 out of 5 billion, whatever. Yeah, that's a misrelease. And all of a sudden you get all these red mosquitoes instead of all these gray mosquitoes. Right now, we got a lot of gray mosquitoes and they're climbing up higher. The bird is faced with gray mosquitoes there. Now you went and switched, but something went wrong and you released a red female. Not released, but something went wrong and you got red females and you got all red mosquitoes. I have no idea what that does to the poor bird. Okay. I have no idea what this does to human pathogens that Culex carries. I don't. And I have no idea if this upcountry stuff, red, is coming down country to make all the mosquitoes in the lowlands red. By the way, Culex, West Nile, and all these other diseases it carries, we got 2 mosquitoes here I worry about, Culex and Aedes albopictus. Albopictus carries dengue, which we fought, carries Zika, and all these things, but Culex certainly carries stuff. So that's what could go wrong. Well, how could it go wrong, Dr. Pang? Because you just showed us that when you cross, here it is, you cross a gray natural female, Wolbachia gray, with Wolbachia red male, it all becomes sterile. Well, so how'd you get your red female that you blew away the population from only red male? How'd you show me the mechanism. Well, it wasn't sexual, it's horizontal. Horizontal means not vertical down the generations, but crossing. And I told you that if you ever released a red female, it could blow up to make everything red. Okay. First of all, if you're going to do that, can you recall this life form? Never mind how the mistake was made. Never mind who you blame. Can you fix this? Oh, could you recall normal mosquitoes? Can you recall fire ant, stinging nettle caterpillar, albizia, ta'api, coqui frog, rapidoia, death, rats, mongoose, prawns? Can you recall life forms? Wolbachia is a life form. Happens to be very microscopic. Can you recall a microscopic life form that's even harder than a life form? Remember that, because they can't say, well, Penn just complains. I'm going to show you something I think you can recall, but that's later. Show me the mechanisms of horizontal gene transfer. Here it is. Here's the female, sorry. Here's the male mosquito with your introduced Wolbachia red strain. I don't know the name. Never mind if you can remember the scientific name. I just call it red strain. Here's your female, naturally occurring with Wolbachia, with a gray strain. How in the world, besides sexually, could they contaminate each other? One, common sugar feeding source. You mean this guy could spit while he's eating, and this one trying to eat the sugar source, same sugar source, flowers, nectar, could pick up the spit of him with Wolbachia and get infected? Yep. Okay. Well, that's odd. So they feed commonly within about 200 feet. That's how far they go for feeding, because if they have to go far, they ain't going to make it. But they could also drifting from the lowlands or whatever, or drifting from the highlands to the lowlands between flights. So the area that they try to feed mutually off of sugar feeding is about 200 feet. But the real drift is farther depending on the wind. But just look at the feeding area, the sugar. So here's your red Wolbachia contaminating all those when they spit and slobber. Oh, wait a minute. I thought you said Wolbachia was obligatory intracellular. How did it come out in the spit? Well, you remember there was an article published that this stuff can survive months outside the intracellular. And certainly it can survive months in the spit. That's not conjecture. I can tell you, I will give you the references because the ants do it. Ants with one kind of Wolbachia, they feed on the fungus. They have these fungus gardens and they slobber. And anything that feeds on the garden, other insects, other ants, same ant, different Wolbachia, they will cross-contaminate each other through the spit. And now anything feeds on the fungus garden, male and female. Okay. Here's another way. One horizontal mechanism wasn't good enough. Well, this mechanism is called serial predation. Something bites your insect, your Wolbachia insect, and it's not quite, well, it contaminated its mouth parts. You can label it green, you can see it green, and then it goes and bites another one. Oh, so it's biting all these things, and it doesn't kill all of them. Some of them survive. And lo and behold, here's the classic example is aphids biting on, wasps biting on aphids. So the aphids got different strains of Wolbachia, red, blue, black, and the wasp is going around biting each of them. A lot of them it kills, but some of them it just wounds, they escape. I don't care if it's biting male or female aphid. Pretty soon you got male and female aphids all cross-contaminated with whatever the thing is spreading, the wasp is spreading. Do you have wasps biting mosquitoes? I don't know. What bites mosquitoes? Here they are, dragonfly and damselfly. Some of our damselflies are in the invasive areas, the endangered areas of the country. I didn't know that. But damselflies and dragonflies like to bite mosquitoes, really, or eat mosquitoes. Where? In the adult flying stage in the air, but more likely in the breeding sites. You mean, hey, how does that go again? The damselfly or dragonfly larva is laid in the same site, small collection of water, bromeliad upcountry, as the mosquito. And you got two larvas, the damselfly larva and the mosquito larva. And one is the damselfly larva predates on the mosquito larva. And just like the adult, it might not eat them all, but it might just bite a whole bunch. So on the aerial side, damselfly, dragonfly bites your red male, then goes and bites a female gray, not sexual, and she escapes and lives. Now you have a red female. We decided red females aren't too good, not when there are too many of them, because it could blow the whole population red. But if you think about it, the guys who were just dying to say, well, you're wrong, Dr. Bang. Because how in the world would you get a red mosquito wiggler? Because all the offspring, egg, to wiggler is sterile. So all the wiggler coming through egg of this cross are sterile. There are no red wigglers. That's true. But you see, the damselfly or dragonfly, it's red. And he laid it female laid it. And now you have red damselfly wigglers, serially predating on male and female gray mosquito wigglers. Okay, yeah, think about that one. Okay, but once again, these hatch and you get red females. Okay, so if you don't think that's true, here's the thing about here. This one, the third one, is the spreading of Wolbachia in ants, common feeding in ants. This next one is the predatory wasp. I don't know about wasps, but I worry about a dragonfly and damselfly doing the same thing, predatory feeding of damselfly, zero predation. A lot of people said, I've already heard rebuttal, not public, that yes, these things will spread. But those things, when they get red, they're weak. They're mutants. They'll die out. Not so. The study in Singapore right here said that the spread of these novel mixed Wolbachias are so widespread. 76% of the insect species, not of a certain species, will be taking over. And so it's not just going to die out. This thing, once it spreads, it spreads. Furthermore, you know that, don't you? I just told you about the dengue control studies where they're trying to spread an entire population to red from gray because a red population of mosquito is resistant to dengue. Well, did it wipe out? Not so good in Brazil after 7 months with 35% red. And then in Indonesia, I think it was 90% red. Okay? So these things can really spread. And then Tina had me put in this, parasite brainwash grasshoppers into death. If you study this article and their conclusion is the parasites are driving the world, not the insects, maybe the viruses and parasites, but this is a microbial world, okay? All right, any questions? Otherwise, I'm gonna move on to alternatives.
I have a question. I don't know if you really went into this, but one of the major risks that we talked about with the Wolbachia was increased pathogen infection, which would be in the mosquitoes, and then I would imagine in other and arthropods. And so could you explain a little bit more what increased pathogen infection means, what the risk of that is, and how the Wolbachia is causing that?
Okay, we don't know. They're studying that. But you remember, Tina, it goes both ways. A Wolbachia coming into a mosquito can make, not the humans, but coming into the mosquito, can make the mosquito more tolerable and promote the pathogen, let's say disease acts, or it can make it resistant. We don't know. But for dengue, they chose mosquito to be resistant to dengue. If you go put in some other Wolbachia, it might make it more susceptible to dengue, then you really blew out the dengue problem in humans. Okay, they don't know. Okay, and I just read a report because I was looking about this. They've got some enzymes and some points of. I do not think it's a straight gene mutation. I think it's a gene expression which allows the pathogen to do well or not do well, depend on the strain of Wolbachia in your mosquito and in your host. But like I said, if you turn the entire upcountry red with your new Wolbachia strain and that blows to the lowlands, I have no idea what red Wolbachia carrying Culex is going to behave regarding West Nile and the diseases they do. Okay. Furthermore, if that thing spreads to the other mosquito, Albopictus, that's dengue, horizontal transfer, mosquito strains to mosquito strains, I have no idea what that's going to do to dengue, Zika, and all those other things. And if you make a mistake, can you recall it? Really, I doubt it.
No, not in nature. Once you start messing with nature, it goes on however it would like to continue to go on.
Yes. Show me you can recall little fire ant first. Now, if you give them the benefit of the doubt, well, look, Patty, is the best. I'm doing this in good faith. You remember the guys in FDA, senior people who said, I do have conflict of interest, but I can put it aside? They were either lying or deluding themselves, okay? So you state your conflict of interest. Don't tell me how you can control it or not. It's for us to decide, gee, the guy has conflict of interest. Maybe he can control it, number one. Maybe he's right. Maybe he will save the birds. But this is quite a risk you're taking because maybe you can't recall this, okay? Okay.
So
And
do you think,
go ahead.
no, you finish what you're saying and I'll ask. I don't want to miss something.
And the generations, remember, we're supposed to transmit to the future generation science, the patient or the public, the general public, and the students. You think when I tell the students, you got to go do dengue on Maui, they're going to say, well, who brought this stuff in? I don't know. And things got out of hand. Well, nobody brought it in, but can you control the mosquito? Show me you can control the mosquito before you bring in a Wolbachia strain.
So I'm wondering if, do
ahead.
you feel like the state has been given enough information about this horizontal transmission risk? Because I asked about that as well at the virtual meeting. And, we keep getting responses if they don't know about it. And I have definitely submitted, the information that you shared with me last year. This was months ago. I submitted it as testimony and directly sent it in certified mail to the DLNR with these documented peer-reviewed studies, and they're still responding. Send us more information. If we don't know about it, we need to know about it. Do they know about this? Are they looking at this? Where do you feel this is?
I think they do, because coming back through the grapevine, I know you gave them the references, so maybe they got to read them. Maybe they're looking for rebuttals to what I submitted. Maybe they can't find it. I don't know. But I've already heard some feedback through the grapevine that they started to rebut that these things are novel to the new introduction and they will not survive. It's not true. The Brazilian guys are trying to overtake the slums with red mosquitoes and the Singapore study. So they're already beginning to rebut this, but they do not seem to want to do it publicly. So I don't know. I mean, if I talk about ivermectin pro and con publicly and they cannot rebut it publicly, well, maybe that's how people get defamed behind the back. I'm sorry, but this is the way I see it, and nobody in government, not even ACLU, will help me on this issue. Okay, so bring it to a discussion publicly. If the moderator will not address certain questions or tell you, well, this is a good question because I can answer that, who is moderating and what is their conflict of interest? Oh, by the way, Scott, you have conflict of interest? You and Tina? Good time.
No conflicts of interest here.
You have conflict of
interest? I can't think of any.
Has anyone threatened to pull your show for discussing things on?
Well, YouTube already did, so.
For this? For Wolbachia?
No.
Wolbachia. Yeah, I think YouTube suspended mine. But now they're questioning, because the FDA said we didn't go after them so hard. We didn't tell the doctors they couldn't prescribe it. We just kind of frowned upon it. You didn't tell the doctors they couldn't prescribe it. No, you just pulled their license and got them fired. We didn't do it. The state board did. What did the state board quote? They quoted you. You're just getting people to do your dirty work. Huh? Come on, enough of this. Okay, good. Okay, Tina, ready? Shall I move on to the alternatives?
I'm ready.
Okay.
Right.
Be constructive, not saying bad things about things if you don't have a good solution. Here's my alternative. Maybe you can stop and recall it. Some people said, no, you can't. I come from a malaria background. I've been doing it for 40 years. I taught for the World Health Organization and published. There are a certain bunch of antimalarial drugs, okay? Some drugs you can treat blood stages. Now, there's bird malaria in birds, rat malaria in rat, mice malaria in mice, human malaria in human. We have our own malarias, but it's warm-blooded. So some antimalarial drugs can treat the blood stages. They probably can treat the warm blood bird stage too. Okay. There's a bunch of other drugs, not a bunch, maybe 3, that treat the mosquito stage. Each malaria has to go through all these obligatory different stages in the warm-blooded thing, maybe 3 or 4 in the human, and maybe 3 or 4 in the mosquito. It has to go through those stages. So it's not one drug treat them all. Some only treat the mosquito stages, and they treat them very well. So if you ever wanted to treat a mosquito, cure its malaria, you treat them with primaquine. There's the chemical structure. How do you make the mosquito drink primaquine? You can put it in sugar feeding, or you can dose the poor bird. Doesn't do too much for the bird, but when the mosquito bites the bird, that'll cure the mosquito's malaria. And lo and behold, I studied the literature 2022, recent. Ivermectin, there's the drug that caused my problem. Apparently that can treat the mosquito stage too. Not all stages, maybe about 80% of the stages, but I think primaquine can treat them all. All right. So the EA, I mean here on the red side, yeah, the environmental assessment said they considered all the alternatives. And in the text, it says, we got 2 alternatives. Do it our way or don't do anything. So I said, that can't be. So I went to their index or their appendix, and they considered some alternatives. Thank goodness, there were 9 alternatives. One was the anti-malaria drugs. I said, okay, here we go. We can overlap on this. I'm kind of a malaria guy. And they said, to use your anti-malaria drugs, you have to diagnose all the birds with malaria, bring them out of the area, they kind of might be half dead already, treat them with anti-malaria drugs, help them recoup, and send them back to the field. I mean, the poor bird's going to be traumatized or dead or whatever. That was their use of the anti-malaria drug. And I agree. If that's the way you're going to use it, then I'd list it way, way, way down in the appendix. The appendix of appendices, because that's just too much trauma, diagnosis, and treatment of the birds. We do that with humans, but we don't do it with. So why can't we just treat the mosquito? Well, they didn't consider that because the original study was 1961, and I guess they weren't born before then. But, well, it wasn't recent. Yes, it was. It was probably republished again as mechanism for controlling human malaria in 2022. Because you don't have the vaccine, do you, for the humans? So can't we just control it in the mosquitoes somehow with these drugs? Hey, why can't you? Here is the mosquito upcountry. It lives a longer time, 20 days. There are more of them, but the ones that do survive the cold, this is it, Tina, I think they're living longer. Sometimes I read reports where they're living shorter, but some are living longer. Look at the parasite, the malaria parasite developing in the mosquito. It has to take 12 days to develop. If your mosquito longevity is shorter than 12 days, all your mosquitoes can get malaria, but they can't give it because the mosquito lifetime is shorter than the parasite development time. And this is humans and birds. So for a while, in the highlands of Africa, they did not have malaria. The mosquitoes had it, but they weren't living long enough for the parasite to develop. As soon as the mosquitoes live longer, or the parasite development time is shorter, now you're having malaria mosquitoes in the highlands, either to humans or to birds. We know this quite well because we had a meeting in 2005 watching human malaria creep up into the highlands. Okay, all right. Here is your mosquito taking blood feedings. It takes a blood meal to make eggs every 3 or 4 days. Eggs of the mosquito. The parasite takes 12 days, maybe. And the mosquito wants to feed and produce eggs every 3 or 4 days. So during your time of parasite development, it might take 1, 2, 3, 4, 1, 2, 3, 4, 5, maybe. Okay. Every time it feeds on a blood meal, which is non-bird. Well, my next slide will come up. That's good because it's not giving it to bird, it's giving it to what's up there, pig. Pig don't get bird malaria. So the pig will dilute out the malaria. Even better, if you dose that pig with primaquine, you cure that the mosquito of all malaria. So when the mosquito bites the bird the next time, bite bird, got malaria, bite pig, didn't give malaria, but the pig not treated with primaquine, bite bird, not developed yet by bird, not developed yet by bird. Now it's developed, and you gave it. I want to cure the mosquitoes of malaria. Check this out. I'm going to blood feed. The blood feed is on the warm blood, bird or mammal. The mammals up there were the pig. Remember the hoof prints making breeding sites. Here's your mosquito, normally cycling malaria in the bird, warm blood. Okay, the parasite takes 12 days. Mosquito lives 20 days. In the last 8 days of its life, it's giving malaria. Okay, but it's always getting malaria. If you were to bring in, never mind the dosing of primaquine or ivermectin now, just look at the rabbit. Just look at the rabbit. If you were to bring up rabbits and the mosquito, instead of biting bird, it takes any warm blood, bites the rabbit, what happens to the rabbit? Rabbits only get rabbit malaria, okay? And it dilutes out the bird malaria, okay? It's called zooprophylaxis. Zoo means animal. Another animal will prevent malaria transmission back into the birds because the rabbit doesn't carry bird malaria. So it's blocked by the rabbit. Really? Is it true? Is it true? This is what we did for malaria before the drugs. So the Eastern Africans, they keep cattle. They live off their cattle, milk and blood of the cattle. Okay. And they noticed that Africa is endemic for malaria. In the bad malaria seasons, the humans were getting human malaria. And they found out, I don't know if they found out or they stumbled upon, that when they drive the cattle under their house, their malaria epidemics kind of go away. Now, you might say, well, why did they drive the cattle under the house? I don't know. Maybe so many human Africans had malaria, they couldn't keep control of their flock. People were stealing them. But when you drive the cattle under the house, the mosquito chose to bite cattle. And the cattle don't hold human malaria. And so without primaquine or anything, it diluted it out. And the British termed this zooprophylaxis. Hey. So I would like to bring up caged rabbits. I looked at guinea pig, I looked at rat, I looked at rabbit. Caged rabbits, don't ask me where, they're lab bred, so they're not bringing weird pathogens in. They're sterilized males. They're all unisex in case they escape and you didn't sterilize them. They're all males. Okay, but I'll sterilize them. So we put them in cages, they don't run around, and we let the mosquito bite them. I will dose their feed, we won't talk about how yet, with primaquine. Primaquine is quite safe, low doses. I'll add in ivermectin. And every time the mosquito bites one of these, not only do you do zooprophylaxis, you cure the mosquito of malaria. Well, where are you going to put these rabbits in cages? I don't know. Shall I put them in the site itself? Are you going to drone in rabbits in cages and drone in their feed laced with primaquine? Well, you got to give them water. Not with primaquine, but you got to give them water. I don't know. How much can your drones carry? Commercial drones, 400 pounds. I don't think rabbits in their feed are going to weigh 400 pounds. Besides, you should spread it out. But if you are expecting drift from the lowlands, I won't even go up there. I'll just put my rabbits on the lowlands out of the protected area because the mosquitoes biting the lowlands will come up loaded with primaquine and be cured of malaria. Okay. So what if something goes wrong? I don't know. Just take out the rabbits, stop your primaquine, kill the rabbits. I don't know. Just stop it. This is not a life form. This is a drug. You're putting drugs in the upcountry environment. This is not a pesticide. This is a targeted drug going into rabbit. If you say you're releasing primaquine in rabbits to these native endemic areas, hey, you got humans going up there. Aren't they on some drugs? Aren't they on anti-diabetic drugs? I mean, there's something releasing a chemical to the environment versus a dose of a chemical into an organism which you can shut down. All right. Enough of that. Any questions on that? I'm not saying it's going to work, but I want to consider.
I've got a couple of questions.
Okay.
I mean, first, I'm worried about the rabbits, and I think a lot of other people would be too. So definitely that's going to be another probably
Cage rabbit,
controversial. Yeah, I don't, it makes me uncomfortable. But I like the idea that there might be a better solution. So maybe there's a different way to look at that. But I'm concerned about what exactly is primaquine and even really ivermectin derived from, and if that gets into animals that are part of the food supply, like the pigs. Do people need to be concerned at all about that, about residuals of those pharmaceuticals?
These are very short-lived drugs. Primaquine, I think it's derived from the chemical industry of Germany. Before the Second World War, they used to do fabric dye, and they pulled out some chemicals, these compounds, which have antimalarial effects. Ivermectin, we should know about. It's given the Nobel Prize. It's derived from a microbe in the soil, used very widely. We know the dose. We know it's quite safe, and it's almost ubiquitous in the soil anyway. Okay, this one, you're right, is artificial, but this one we've used extensively and widely in humans since 1940. We know the dose. We know it's quite safe, but I can recall it from your environment, except if you say, well, what if the birds eat the primaquine and ivermectin? That's why it's a cage animal with grain. The birds are honey-free birds. I was going to put the rabbit food in the back, but we can talk about this. The point is, Tina, I don't mind you asking these questions. I don't mind the agency asking the questions. Where was this alternative surfaced? And I don't mind sinking the alternative, but I want to see it considered. Okay.
Maybe we can all just take ivermectin all the time and let the mosquitoes bite us, and then we can eradicate the mosquitoes through.
Hey, you laugh.
I actually just read an article on that yesterday.
No?
Actually, how.
Here's one for you. I didn't include this I didn't give references on this. In the Galapagos, they have endangered species, yeah? Yep. They have the same problem, yeah? Invasive mosquitoes carrying malaria to their endangered birds, right? Hey, their mosquitoes have carrying kind of a lot of bird malaria. But how come their birds aren't having a problem? So this is published. They looked at and saw what their mosquitoes that feed on birds, what do they feed on? It's like 95% human. So you have the zooprophylaxis effect. Humans don't get bird malaria without dosing the primaquine and ivermectin. Scott, if you want to say, hey, are you guys in the Galapagos? You're already helping us by zooprophylaxing the birds. Why don't you take some ivermectin? Cure the birds that happen to be carrying bird malaria. Yeah. So guys are proposing, let's see what the mosquitoes are feeding on. Some feed on goat, some feed on pig. And they are diluting out the bird malaria in bird. By the way, Department of whatever, they said that the invasive pigs up there in the highlands, their hoof prints are breeding sites for the mosquito. Fine, eradicate the pig. Did your malaria outbreak increase in the birds when you eradicate the pig? Was the pig also being a zooprophylactic effect besides hoof print and breeding site? Huh. Yeah. Show me you even at least considered that. I don't want one-sided arguments on this. Okay. You know what I'm saying, right? I mean, maybe the pig had a good side to it, making all the hoof prints, but being the zooprophylactic target.
Absolutely.
So everything is on the table, unless you're going to censor Scott and backstab me for whatever. Wolbachia misinformation. Okay, ready? Final slide. No pictures, but I think I can explain this. Ready? That's my alternative. Show me it was considered. The math model. I said I wouldn't mess with this, Tina. I said I take for granted if you cross the male and female, red male with gray female, all the things are sterile. Already people said, oh, but there might be a female in there. You know, misclassification. I can deal with that. There's misclassification. 1 out of a million, 5 out of a million, 1 out of a billion. And we spend all our time about misclassification. First of all, I spent a lot of time on horizontal transfer to create red females. Now I want to spend my time on mathematical model. You introduce n number red males into the area. What percent of the males are red after, I don't know, a month or something? I mean, how much do we have to introduce? Already, it's kind of appalling. You're going to do millions a week? How much is too much? I have no idea. How much is enough? I don't know. Okay. You can calculate the percent of red males in a closed system. But from what I read, they got to give me the full text. Their model did not allow for drift from the untreated areas. So your percent males will drop. Some guys actually say, what if your red males blow out of the area? I don't know, that drops it even more. So what is the distance the males mosquitoes spread? I don't know, on the wind, on the sugar feeding, whatever, between sugar feedings. Fine, do what you want to do. Do your MRR study. Before you dose them with Wolbachia, you dose them with some kind of color paint or a marker. So you release N marked males, and after a certain period of time, you can say, whoa, X% of them are my marked ones. We can back off. We don't have to release so many, or not enough. We got to release more. That's your model for population dispersion. There is another model here, many models, and I mess with models, and I continue to do and publish. And it's called the choke point, or the rate limiting step. Okay? It's also called a bottleneck. Somewhere in all this I was reading, somebody said, why don't you have more mosquitoes up country? Why is it limited? What's limiting them? For the cold temperature, that'll knock out half. Yeah, but if it knocked out half, why don't we have 500 instead of 15? Somebody said the choke point was the breeding site, that there's limited number of small collections of water, which will remain wet for 10 days, that the thing has to hatch. And that's the choke point, okay? I thought it was sugar feedings, 200 feet. Where do they feed? I don't know, fruit, berries, flowers. This is up country. I don't know how much sugar is up there. But then I said, okay, let's look at a choke point. Gotta name it. All right, here we go. Even if you know the percent on the MRR study, you have to deal with the rate limiting step in your model, also called choke point or bottleneck. It could be breeding sites, it could be sugar meal. I'll give you one example of a choke point example, and you tell me if this doesn't apply or not. You ready? People have said, don't do this. It's politically incorrect. I don't care. There's an open border. I don't care what border it is. Maybe it's the Ukrainian border. Maybe it's the southern border. There's an open border, okay? You have a control site at your border. You can allow 200 people per day to pass your choke point. It's not the entire border. It's your facility. Why do you only allow 200 a day? You got to do drug screening. You got to do COVID testing. You got to do background check. So I can only do 200 a day, okay? But how many people are challenging my border? 2,000, okay? 2,000. And I'm only going to let through 200 a day based on my criteria. You come up with an intervention that can cut 2,000 people challenging to 1,000. I can cut it by half, okay? For example, all these people are challenging the southern border. Somebody says, well, I'm going to pass a new rule. Venezuelans and Haitians and Cubans, they won't come in one way or the other. You're going to cut them by half or not. You could be Asians. I don't care. You cut the people challenging the border by half. So only 1,000 challenge the border, okay? Well, how many is still coming in? Well, it depends on your 200 per day gate, yeah? If you exclude race before they come to the gate, still 200 come in. They just happen to not be of the race. But if half of them got to go, they're the wrong race, then you affected the population. Got it? So when you choke off a population by Wolbachia sterilization, did it occur before the choke point or after the choke point? And if the choke point is breeding sites, which somebody told me it was, and Wolbachia renders them sterile, like they're not going to lay eggs, do these females that would have laid eggs sterile compete for the breeding site, in which case you will decrease the population? Or do the mosquitoes say, well, I'm not going to lay eggs, I won't challenge the breeding site. And now you still have the same number coming into the breeding site, and they happen to be all non-red, okay, all non-affected. So that's how you model on the choke point. And I would like to know, I'm not going to look this up, tell me, when you introduce and crossbreed a red and a a red male with a gray female and render her sterile? Does she still compete for a breeding site? If she does, you control the number coming out hatching. But if she doesn't, you still have the same number coming into your breeding site and breeding. Okay. Oh, by the way, these mathematical models we're publishing now, they're called ordinary differential equations. I would like to see it. Maybe some people don't mean much, but I do believe I got the A+ in Princeton mathematics, third year mathematics in ordinary differential equations. There was only one given to me against the math majors. So I'm not trying to brag, but do not defame me unless you're willing to go with me fact for fact and equation for equation. Sorry about that. Okay, that's all I have to say. Open it for questions.
I've got a few questions that have to do with the math because we've been crunching some numbers based on the science that we have found on some of this information and then some of the documentation from the state and the EPA as far as the female release, which that doesn't even take into account the horizontal transmission factor. But the first thing is that you had the lifespan of the mosquito is 20 days, and we have documented that the females can live up to 8 weeks. So that's a possibility, right? But that's not necessarily what might be happening in the environment here, but it could happen. 8 weeks is like the high end of the female.
Yes, that's even worse, right? They live longer. You get more chances for mosquito broods to finish, but then that pushes the primaquine, pushes the rabbit model because you get more chances to kill off any one of those. The more it lives, the more mosquitoes it's going to make, but the more chance you have to sterilize its mosquito carriage.
Okay.
So remember, Tina, it's the ratio or the comparison of how long the mosquito lives by how long the malaria takes to develop. It's always that interplay.
And then your numbers were showing a breeding factor, like how many offspring. I'm not sure how that correlates with, we're looking at that one female mosquito can produce 20 female mosquitoes within 2 weeks as a starting point. And that's a low end estimate being conservative. That's accurate
Yes.
compared with your numbers as well?
Okay. I ran into different multiplication factors. Yeah. So on one breed, it might do 20 to, let's say 100. Then all of a sudden I'm reading and it goes like 3,000. What's that? That's in 2 or 3 breeds when they make more generations. Okay. So you're right. I pulled it back to this because if you say, well, the mosquito increased 5,000 fold, that's over like 4 generations, 20 times 20 times 20 times 20, whatever. So you're right. We have to sit down slowly and go over the multiplication factors, okay? But don't forget, before we even address that, I want to know of introduction of drift, okay? Because forget the Wolbachia. You diluted out my Wolbachia. Whatever you did to the mosquitoes, more are always going to blow in. And the bottleneck is what? How many blow in or how many they freeze up there? Or it's going to get a little messy, but I agree with, you gave me a basic quote. A lot of guys say, we don't really know what's going on here. Oh, by the way, if you did know what's going on, eradicate the mosquitoes without bringing in Wolbachia. Show me you can do it.
Yeah.
Right.
Absolutely.
They've even admitted that they don't even know if this will work in the federal documentation for this series of projects. Did
you bring that up with the EA moderator and say, why don't you address that question?
Yeah, I think that may have been asked, not by me, but another question along those lines. But I'm not positive. But I do know that it is in the federal documents from the Department of the Interior tied to this project, and it says we don't. This has been done with humans, but we don't know if it'll work. In think in the wild environment.
And what did the local guys, facilitators, say about that? What they say is true. We know more about this in the slums of Brazil and Indonesia for dengue because we're trying to expand the mosquito model. But you don't know it that well because how come in Brazil, the takeover of red mosquitoes is 35%, and in Indonesia it's 90%? you're trying to make it 0, and you can't even pin down how much it expands when you want it to expand. But anyway, okay, so, but you're right.
Yeah. And then the last question that I have, and I don't know, this is more just to see if maybe you've seen some information documented that I haven't, but as far as the rate of accidental release of the females, the state's own permit application to bring these mosquitoes in states that rate as 1 female per every 250,000 males. And that's also what it says on the EPA website in regards to Mosquito Mate, which is one of the labs where they would be bringing the mosquitoes in from. So 1 female for every 250,000 males. And the state is now saying, because of technology and some study that they've seen, that it's virtually impossible that this is going to happen. And they have some different figures that they're putting out there. And I haven't seen any documentation on that specifically from the state. So have you seen anything where we can verify those new numbers?
I saw what you sent me and tried to read behind it. Let's all keep it per million. So if it's 1 for 250,000, that's 4 per million. Then you can say, well, how many are you going to release? Like billions. Okay, whatever. Then I also saw that other thing. We can drag this down to 2 per 100 billion or whatever. Number one, I could do that on a double read. You give me everything you're going to give, and I'll read it twice. Then they said, oh, we're going to use artificial intelligence to read it, blah, blah, blah. Bill Gates going to get involved in artificial read. You're right. I wish it would just pin the number down. It's either 4 per million or 4 per 100 billion. And how are they going to do that? But before we get really into the detail, what it is, how about the incoming mosquitoes and the horizontal? I'm trying to get a balanced approach. They will spend all their time addressing the things they have the better handle on. When in fact, I think like horizontal transfer, can you please tell me about that? Because, common sugar feedings, mosquitoes slobbering all over each other's, the thing living outside, the mosquito for a long time, infecting each other. Wow.
Yeah, and
worrisome.
So we have this opportunity, the public, to comment on the environmental assessment, which there's a deadline of January 23rd. And if everyone can go to the website, hawaiiunites.org, you can find a link to comment on that. But what I would specifically want to say is, this is why we need to demand an environmental impact statement and far more detail on, these risks. They haven't addressed the horizontal transmission at all in the environmental assessment, and they haven't addressed the accidental female release and these figures, the one that is already documented through the state's permit and the EPA, and the one that they're now discussing. None of this is in the environmental assessment. So I think people need to speak up about that and question, why we don't have more details and why this wasn't studied much more seriously with the effect that it have on the environment, the public health, the birds, and everything else. So I
hate to say this, but it wasn't studied so seriously because it's only an EA. And now you have a catch-22. Forget what it's called, EA, EIS. Can you please study this seriously if you cannot recall what could go wrong?
It's irreversible, is what you've said. This once they do this and release these mosquitoes, this is irreversible.
Yes. Now, if they're going to say, well, then what gives me the right to say rabbits are more reversible? You're right. I could kill the rabbits and remove them. Okay, I can remove the primaquine from their feed. Okay, but I feel better about that than letting Wolbachia get out of control and spreading from the highlands to the lowlands and forever being in the highlands.
Yeah, that's something that's absolutely not controllable. That's out of anyone's hands at that point once you release it into the population.